Bismillahirrahmanirrahim, and welcome back to this next video. And this is the last video in the series of videos on the hemostasis. Now, in my previous videos, I told you about the hemostasis, about the primary hemostasis, and one type of the secondary hemostasis, that was the extrinsic pathway of the secondary hemostasis. So, the first thing is, what is secondary hemostasis? Now, the secondary hemostasis, it actually refers to the cascade of enzymatic reactions that is carried out by the clotting factors. So, the secondary hemostasis would be carried out by these clotting factors, and the ultimate result of these enzymatic reaction is the conversion of the fibrinogen to the fibrin monomers. Now, what these fibrin monomers do, they are cross-linked into insoluble strands that serve to stabilize the loose platelet clot that has been formed in the primary hemostasis. I have a detailed video on the primary hemostasis, and I'll share the link in the description. Now, the secondary hemostasis, the one that we are talking about now, is of two types. One is known as the extrinsic pathway, on which I have a detailed video, for which I'll share the link in the description. And the second one is the intrinsic pathway, which is actually the focus of this particular video. So let's talk about the intrinsic pathway. Now, the intrinsic pathway is also known as the contact activation pathway, and this is the longer pathway of the secondary hemostasis. It is longer because the number of clotting factors that are utilized in the intrinsic pathway, their number is more as compared to the number of the clotting factors that are used in the extrinsic pathway. So this one is the longer pathway of the secondary hemostasis, and the extrinsic pathway is the shorter pathway of the secondary hemostasis. So let us define these terms again. What is hemostasis? Now, this hemostasis, it actually refers to the cessation of blood loss from a damaged vessel, which is followed by the repair, so that the blood loss that can be stopped. The second thing is, if there is secondary hemostasis, of course, there should be a primary hemostasis as well. So the primary hemostasis is actually the formation of a platelet plug at the site of injury, and the secondary hemostasis is actually the addition of the clotting or the coagulation factors to stabilize the platelet plug that has been formed by the primary hemostasis. As the plug formed by the platelet is a weaker one, that is not a stable one, so the secondary hemostasis is going to stabilize that particular platelet plug. So when there is damage to the blood vessels, let's talk about in more detail about the intrinsic pathway. So when there is damage to the blood vessel, there is exposure of the subendothelial collagen. So on the interior of the endothelium or the endothelium cells, they have got these collagens, and when there is damage to the blood vessel, the subendothelial collagen that is going to exposed. Now, when this subendothelial collagen get exposed, there are attachment of three important things to this subendothelial collagen or to the damaged blood vessel. The first one is known as the factor 12, which is actually a protein, and this is a serine protease. The second thing that get attached to the subendothelial collagen, that is the prekallikrein, and this prekallikrein is also a serine protease. Don't worry about serine protease, I'm going to define that for you in a while. The third thing that is going to attach, that is the high molecular weight kininogen, and this HMWK is actually a circulating plasma protein, and what it do is that it is serving as a cofactor for the activation of the prekallikrein into the kallikrein, and the factor 12 into the active factor 12. So what happens is that in this particular complex, when these three things they get attached to the damaged blood vessel, the factor 12 is going to be converted into the activated factor 12. So if you can see over here, there is no small a, there is a small a over here, and this small a actually refers to the activated form of a particular clotting factor. So this is the inactive factor 12, this one is the active factor 12. So how this factor 12 it gets activated? It is actually activated by the polyanions which are there in the bloodstream. The second thing is, when this factor 12 it move into its activated form, so the activated factor 12 is going to act on the prekallikrein, which is the inactive form of the kallikrein, and when this factor 12, the activated factor 12, when it acts on the pre-kallikrein, that is going to be converted into the kallikrein. So the function of the kallikrein, what it do is that it actually helps in the vasodilation, so that the clotting factors that can easily reach to the damaged blood vessel. So we have discussed the function of all of the three things that are attaching to the damaged blood vessel or the subendothelial collagen. The HMWK is a cofactor for the activation of these two things. The factor 12 is going to activate the pre-kallikrein into the kallikrein, and the kallikrein is actually helping in the vasodilation. So when the factor 12A it gets activated, it also serves another important function, and that is that this activated factor 12, it acts on the factor 11, and when it acts on the inactive factor 11, the factor 11 is going to move it to its active form, which is the activated, like this small a is over here, so this is the activated form of the factor 11. Again, this factor 11 is a serine protease. So when the factor 11 it gets activated, it is going to act on factor 9, and when it acts on factor 9, the factor 9 is going to be converted into its active form. Again, this factor 9 is a serine protease, so we are talking more about the serine proteases. So when this factor 9 it gets activated, it is going to combine with its cofactor, which is the activated factor 8. So what you can say is that from where this activated factor 8 comes. Initially, when there is no damage to the blood vessel, this factor 8 is in complex with another factor, which is known as the von Willebrand factor, which is again a glycoprotein. So the factor 8, when it is in combination with the von Willebrand factor, it is in the inactive form. So when we need the factor 8 for the activation of the factor 9A, what happens is that the factor 2A, the activated factor 2, that is going to act on this particular complex, and when it acts on this particular complex, the factor 8 is removed from the von Willebrand factor, and the factor 8 moves into its active form, which is the factor 8A form. And when this is activated, it is going to bind with the factor 9A, and it will serve as a cofactor for this. So the factor 9 with its cofactor is going to perform its function now, and its function is that this particular complex work on the factor 10, and when it works on the factor 10, the factor 10 is converted into its active form, this A form. Now when the factor 10A it gets activated, it is going to combine with a lot of things. The first thing it is going to combine is the platelet membrane phospholipid, for short that is known as the PL. It is also going to combine with this factor 5A, this activated factor 5A. Again, the factor 5 move into its activated form when it is acted by the thrombin or the factor 2, and it is also going to combine with the calcium. So the factor 10A, the PL, the activated factor 5A, and the calcium, when they combine with each other, they are going to make this particular complex, which is known as the prothrombinase complex. Now this prothrombinase complex, it's going to act on the factor 2, which is the prothrombin. Actually, this prothrombin is the inactivated form of the thrombin or the inactivated form of the factor 2. So the prothrombin, which is produced by the liver, when it is acted upon by this prothrombinase complex, it is going to move it into its activated form, which is the factor 2A form. This thrombin again is a serine protease. So let me define what the serine protease mean. Now the serine proteases, which is also known as the serine endopeptidases, they are actually the enzymes that cleave the peptide bonds in the protein. So they are proteases. Why they are called as the serine proteases? Because the serine amino acid, it serves as the nucleophilic amino acid at the enzyme's active site. Therefore they are serine proteases, a specialized class of the proteases. So when the factor 2A it gets factor 2A, it acts on the inactivated factor 1, which is the fibrinogen. Again, this fibrinogen is produced by the liver. So when the factor 2A it acts on the inactive factor 1, the inactive factor 1 is converted into the active factor 1. Now this active factor 1 it acts on the it is going to combine with the factor 13, and when the fibrin 1 it combines with the factor 13, the factor 13 is going to move into its activated form. And this factor 13 is actually the fibrin stabilizing factor. What I mean by that is that the fibrin, the factor 13A, when it combines with the calcium, it is going to make this fibrin into a stabilized form. Let me show you this, how this factor 13 is actually acting as a stabilizing factor for the fibrin. As you can see over here, this cross-linked structure over here, or this cross-linked fibrin plug, that is actually a stable plug. What happens is that when these fibrin that move into their activated form, they are going to make these a longer strand on these polymeric structure. But this polymeric structure, when they are in in this particular form, this is a very weak kind of the structure. So what the factor 13 do is, it is going to cross-link these fibrin polymers to each other to give you a stable fibrin plug. And you can imagine it like this. This is going to be the factor 13. So what they are going to do is, they are going to you can say cross-link these fibrin polymers to give you this stable fibrin plug, which helps you in the stopping of the bleeding from the damaged blood vessel. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.